In this video, I'm going to talk to you about the history of early life on Earth. Initially, the Earth had no life. Scientists believe it had a reducing atmosphere that was lacking oxygen, and there was an abundant amount of energy sources, UV light, lightning, and volcanic activity. Those conditions set up what was needed for abiotic synthesis of organic compounds. In a famous experiment, Miller and Urey set up an apparatus looking like this, where they essentially created a closed system with water to represent the early oceans and gases to represent the early atmosphere. So remember, no oxygen. And they used electrodes to stimulate lightning. And what they found was that the lightning and those gases, the energy was able to combine those inorganic molecules to form organic molecules. And they were able to produce amino acids, sugars, lipids, and many other organic molecules. In other experiments, scientists have found that when you take a liquid concentrated with hot monomers, so really warm kind of organic compound soup essentially, and with repeated kind of evaporation and cooling, you could actually form polymers. And so they splashed what they hypothesized would have been kind of this primordial soup, this kind of water containing lots of organic compounds, and they splashed them on hot lava rocks and things like that, and they were able to form polymers. So once you had polymers, you could then potentially aggregate them into a cell-like structure. The first cell-like structures are called protobions, and they're basically lipid bubbles. And they set up some of the important basics of a cellular form. So the first thing, because they have a lipid layer, and we know that certain molecules have difficulty passing through lipid bilayers, they were able to create kind of separate internal and external environments. And because the internal environment had a certain concentration of molecules, it was able to establish essentially an early simple metabolism, as shown here. Another thing is because membranes, phospholipids, are fluid, they are allowed to pinch off and they essentially can form other lipid bubbles. And so this kind of idea of exocytosis is essentially early reproduction. Now, to become a true cell, you would need a genetic material. Scientists hypothesize that RNA was the first genetic material. And it's likely because of the fact that RNA can do so many jobs. RNA can act like an enzyme, and it's able to replicate itself. Okay. The other thing that RNA can do is RNA directs protein synthesis, right? tRNA, messenger RNA, rRNA, okay? They're all involved in protein synthesis. So RNA does many, many jobs needed of genetic material. So our very first cells most likely had RNA as the genetic material. But over time, DNA became the master molecule, genetic material, because DNA is much more stable. And so if a code for essentially a beneficial trait is found in a cell, because DNA is stable, it is more likely to be copied correctly and passed on to the next generation of cells. RNA, being single-stranded, is more likely to be kind of damaged and have mutations. And so it doesn't necessarily allow for this kind of continuity of inheritance. So this picture right here kind of summarizes that fact. We're starting off with our inorganic molecules, okay, through heat and lightning and things like that. In our early atmosphere, they were able to form monomers, as in the Miller-Urey experiment. Those monomers were then able to aggregate into polymers, and then our polymers were able to assemble into cell-like structures that had three key things of cells: compartmentalization, replication with a genetic material, and then metabolism. And then that early life was able to divide and over evolutionary time, create all the different types of organisms on Earth. Now, one other thing we need to talk about is this idea of metabolism. So as these early cells were being established, they're able to do different types of chemical reactions in their cytoplasm to obtain and use energy. The first cells were likely heterotrophs, and they were able to obtain energy from the abiotically synthesized organic compounds found in their environment. So those would be the ones created similar to those in the Miller-Urey experiment. Now those first cells, when they took in these abiotically synthesized organic compounds, they would have relied on anaerobic respiration, so glycolysis and fermentation, to obtain energy from these food molecules because there's no oxygen in the atmosphere at this point. Now, this anaerobic respiration can produce carbon dioxide, and there may have been some mutants at the time who were able to then take that carbon dioxide and use photosynthesis or chemosynthesis to then make organic compounds. So at this point now, we're going to have autotrophs beginning to evolve. Okay, so by chance, there were some changes in the genetic code that allowed some cells to potentially do photosynthesis or chemosynthesis and make their own organic compounds. Now, natural selection is going to favor these autotrophs because they can make their own food, their own organic compounds, and not compete with heterotrophs for organic compounds made through abiotic synthesis. Okay, so the heterotrophs are basically have to obtain their food source from their environment, and so they're competing for it. So autotrophs would have an advantage. Now, photosynthesis would produce oxygen, and this oxygen production completely changed the way the Earth was set up. With oxygen, now aerobic respiration would be favored, because if you can do aerobic respiration, you would produce many more ATP per organic molecule. So think about aerobic respiration with mitochondria, they're able to produce like 36, 38 ATP per glucose, whereas typically with anaerobic, they're getting 2 ATP. So that extra energy is going to allow those cells to be able to function higher, be able to do different processes faster and potentially better than their anaerobic competitors. And so evolution would have favored aerobic organisms. Now, once we had these prokaryotes that were able to do both aerobic respiration and photosynthesis, we can then have the evolution of eukaryotes. So the first step in eukaryotic evolution would be the infolding of the membrane to create the endomembrane system, things like the nuclear envelope, endoplasmic reticulum, Golgi apparatus, vesicles, things like that. And that extra compartmentalization inside the cell would have allowed for more efficient chemical reactions and more efficient tasks within the cell. Now, through engulfing other prokaryotes, this early eukaryote could then potentially gain additional functions to help it survive and reproduce. So it is believed that when early eukaryote engulfed an aerobic heterotrophic prokaryote, okay, that prokaryote lived inside its host cell in symbiosis. They became so dependent on each other that eventually that aerobic prokaryote became a mitochondrion that could be passed on from generation to generation. Okay, that ancestral heterotrophic eukaryote could have potentially also engulfed photosynthetic prokaryotes. And photosynthetic prokaryotes would then be able to live in symbiosis in their host cell and become chloroplasts. So that is the endosymbiotic theory of how mitochondria and chloroplasts evolved. Now, the final slide is just to kind of help you put into context of when all this was occurring. Okay, so our unit on this is GAs, which represents 1 billion years. Okay, so early on, okay, we're seeing that we had this early conditions of Earth where we have that reducing atmosphere. There's no life yet. we have lots of lightning and volcanic activity and things like that. Okay, about 3.7 billion years ago is when we saw our first prokaryotes. So remember, those would have been anaerobic prokaryotes, most likely. Early on, there may have been some primitive cells before that, or cell-like structures before that, but that is our first fossil record of prokaryotes. As time goes on, these anaerobic organisms eventually photosynthetic organisms would be favored. So about 2.7 billion years ago is when we have cyanobacteria. Cyanobacteria are photosynthetic bacteria. So photosynthetic prokaryotes, they would be the precursors then of chloroplasts as well. About 2.5 billion years ago, we're seeing the first eukaryotes. So that would have been those with the infolding and the production of a nucleus. But notice it takes about another billion years before we have evidence for cells with mitochondria. And then a billion years ago, we saw cells with chloroplasts. So we're kind of setting up this evolution of having eukaryotic cells, which could then potentially lead to multicellular organisms that are able to do many different functions due to their diverse structures.